Right, okay, so continuing on the subject of multidimensional NMR, we will have the briefest of introductions today into how protein structures are determined with NMR spectroscopy. That is a giant topic with huge books written on the subject. I have a few DOI links to those books in the handout, but essentially the method relies on a collection of highly specialized three-dimensional NMR sequences. And so the first thing we need to do, there she is to introduce three-dimensional NMR spectroscopy. So item 1, 3D NMR experiments. And the principle is largely the same as what we had in two-dimensional NMR, that is, when we have three independent types of nuclei, for example, proton, carbon-13 and nitrogen-15 in proteins, what we can do is make the following general type of NMR experiment. So some kind of preparation sequence, multiple pulses perhaps in there, then evolution time T1, then another preparation sequence, and then evolution time T2, then another preparation sequence, and finally the evolution time T3, during which the magnetization is detected. Now, we cannot, of course, simultaneously vary all three times. We have only one temporal dimension in the space-time that we live. However, what we can do is do real-time detection here and simply just repeat the entire experiment multiple times for different values of the T1 and different values of T2. Now, what we are then going to have is a three-dimensional free induction decay, something like that, where we are going to have the T3, for example, on that coordinate, T2 on that coordinate, T1 on this, and we simply just record FID by FID, value by value, for all of these different direct and indirect time dimensions. And then when we do the three-dimensional Fourier transform, so 3D FT, we are going to have a three-dimensional spectrum. So in the case of proton, carbon and nitrogen, this is going to be something like carbon-13 dimension here on the vertical axis, then proton dimension here on the horizontal, and then 15N on the depth dimension. Turns out that simply just plotting, little clouds in three dimensions here is not terribly useful. Hard to visualize, hard to understand. And so what people do is they do slices of these three-dimensional volumes. Typically, in the case of protein NMR spectroscopy, nitrogen-15 slices. So if we slice it up for a particular value of the nitrogen chemical shift, for example, this one here, then we can extract a two-dimensional NMR spectrum out of that by just cutting this cube and extracting the corresponding yellow plane out of it. And then if we plot that yellow plane separately, it is going to be related to a specific nitrogen signal, and then we are going to have 1H and 13C. And then finally, of course, we are going to have different signals present in the proton dimension. Very few, actually, because we've only just selected perhaps a tiny number of nitrogen nuclei that sit in that chemical shift. And then we are going to have a few proton signals here, here, and here, and there will be the corresponding carbon signals at some points in these various locations. And so if you look at the modern protein NMR processing software, this is how it works. It records the 3D, well, or rather reads the 3D FID from the instrument, does data pre-processing and Fourier transform, and then displays these slices in a user-friendly way. Okay, now to put it into practice, so item 2, protein NMR. We need to draw ourselves a chunk of the protein backbone and take a look at the J-couplings there. Remember that most of the magnetization transfer experiments require J-couplings to exist for the magnetization to move around. So we've got NH, then C alpha and H alpha, and that's coupled to CO, that again to NH, that again to C alpha, and there will be another H alpha and perhaps a few other things in here, so going onwards to some side chain. Then the C alpha is going to once again be connected to CO, and so on. So the protein structure continues. Let me draw some pertinent J-couplings here. Firstly, the one that is quite important is the one-bond J-coupling between the proton and the nitrogen 15. So these are all magnetic nuclei. I'm assuming that the protein is isotope labeled. So this is roughly minus 90 Hertz. And all of this is going to be in Hertz everywhere. And so it is quite easy to move the magnetization from the protons to the nitrogens there. Then there is an important coupling between NH and C alpha. There's one this way, so from there to there, and that's roughly minus 8 Hertz. But then there's also, of course, the direct N to C alpha coupling there, and that is about minus 11 Hertz here. Then we want C alpha to CO, and that is about 50 Hertz. Then CO to N, and that is roughly minus 14 Hertz. Notice that some of these J couplings are negative, and for one bond J couplings that is because the magnetic gyric ratio of nitrogen 15 is negative, and the reason for that is that there's an unpaired neutron in nitrogen 15 nucleus, and that in combination with the intranuclear spin orbit coupling gets you the negative magnetic gyric ratio, and then therefore the negative J coupling. So N to C alpha is once again minus 11, and then, okay, let me see if I missed anything. Well, we'll add them as necessary. Now the experiments in question are labeled by the magnetization transfer path. So item A on our agenda today is the HNCA experiment. And HNCA means that we start on protons, we move the magnetization to nitrogen, then we move it to C alpha, then we move it back to nitrogen, then we move it back to proton, and then we detect. The reason for that, why we start from proton and finish on proton, was discussed in the previous lecture. The sensitivity is much higher and the available magnetization is much higher on protons, and so it makes sense to start and finish there, assuming we can of course have sufficiently efficient magnetization transfer. Okay, and I will only discuss one of these sequences, so I will discuss HNCA in detail, and the rest of them would simply be clear from the abbreviation of the pulse sequence what it does. And it's literally just the sequence of the same inept stages as we have discussed already a couple of times. So we have our proton channel, we have the 15N channel, the 13C alpha channel, and then 13CO channel. And the reason why these are independent is that they are sufficiently separated in chemical shift that we can pulse them independently. And the reason for that is, of course, CO has much of the electron density drained away from it by the oxygen, and so the chemical shift of this is quite high. And then C alpha has a proton next to it and has some aliphatic side chain, and so that chemical shift is quite low. And so by doing sufficiently educated, sophisticated pulses, we can influence these nuclei independently. For practical purposes, they are separate radio frequency channels. Okay, so starting the sequence, and I've got the complete schematic in your handout with all the phase annotations and the sources. Remember what we had to do. We make this magnetization transverse, allow it to evolve for the two-spin order to emerge, and then use radio frequency pulses to flip the spin order from YZ into ZY, then wait for a little bit more, and it all ends up on the nitrogen. So the standard transfer sequence. So we've got the 90 here, we've got a period tau, we have the 180 to refocus the chemical shift, then we have the 90 here, then likewise on the nitrogen. Extra pi pulse is needed here to keep the J coupling going so that it doesn't refocus when we refocus the protons, and then another 90, and we are on the nitrogen. Notice that we are not actually evolving the proton by a variable time. We will now evolve nitrogen by a time t1. Or rather, we do t1 over 2, and then we need to decouple everything else. So we put a pi pulse here on the protons, pi pulse here on the carbon alpha, and pi pulse on the carbon O. And then an extra t1 over 2 period, and that is our evolution. So that's the t1, and at this point all the magnetization is on nitrogen 15. So our t1 dimension here is going to be nitrogen. Then once we are on the nitrogen and we have evolved sufficiently, our next stage is C alpha. Let us move it there. Let us, however, take a look here. We are sitting on the nitrogen and we are now moving to C alpha. So our stages are, we begin here, we move here, and now we enter another evolution period where the interactions will kick in, so that one and that one to C alpha. And CO we will eventually decouple, but notice that's minus 11 and that's minus 8, they are quite similar. So what's going to happen next is it's going to move not just to this C alpha right next door, but also to the neighboring C alpha over here. And so that's the current amino acid on which we are sitting, right? So the nth amino acid, so that's the nth, and that is n minus first. So we have it moving inside the residue, but we also have it move backwards one step. Okay, but here we just wait for a certain period of time, and that's called delta. So I continue this diagram. We have a period delta, and then in here it's just a complicated refocusing game so that these protons do not influence our dynamics. On nitrogen 15, once again we need to refocus, but on the protons it is again a little chunk of the on the carbon it's a little chunk of the inept sequence. So that moves the magnetization forward here. Then we evolve for a time T2 over 2, T2 over 2, so a total of T2. So our T2 dimension is carbon alpha. And finally, we want CO to stay out of the way, and so we put a spin echo on the CO as well. And that is our evolution T2. Right, okay, so we are now on C alpha, but in order to detect, we need to move back to this proton over here, and that is the extra J coupling that I need to mention in here. That is C alpha to NH. It is quite small, so this one, so 2 to 8 Hertz depending, so we cannot really move back there directly. So these two extra pulses, wherever they are, this one is intended to move us back to the nitrogen. And then finally what we have here is, after the period delta, when the move is accomplished, so another delta, we move back from nitrogen to the proton magnetization using a mirror of the sequence that we had there. Tau, tau, and then like that. And finally we detect. Here it's the detection, here it's the decoupling. We don't need to decouple C alpha and CO because they are not, at least not strongly, directly coupled to those protons. So that just keeps on going. Okay, so the schematic therefore is, we start here, we move to the nitrogen, we evolve for a while on the nitrogen, we then move to carbon, we evolve for a while on the carbon, then we move back to nitrogen, back to proton, and we get detected on the proton. A complicated sequence, but let us think about what it would do for us. I've given you a schematic in figure 3 of the handout of this picture. Now, out of our three-dimensional experiment, we would go through the list of all peaks in the nitrogen dimension, and of course, if we look at only the backbone, every individual nitrogen signal corresponds to a separate amino acid. Okay, and then inside the amino acid we are going to have proton signals, and those are all amide protons. They have a distinctive chemical shift because they are bonded to the nitrogen, so they are quite far away from everything else. So this we don't need to worry about, they're well separated. And so all of these nitrogens will be the amide nitrogens, again, one per amino acid. And then since we've correlated them to C alpha, what is going to be here, if we set the chemical shift window correctly, is just the C alphas. And we are going to have two of them per line because this transfer happens into the same C alpha and also into the C alpha of the preceding amino acid. So what is going to happen there, and this is how it's typically displayed. So we have our 13C chemical shift dimension, so roughly between 40 and 70 ppm here. Then we have the proton dimension, and it is customary to cut the spectra into little strips like that, not necessarily in the order of the proton chemical shift, but as you'll see, in the order of the amino acid sequence. So let's say we know, and eventually we do, we can just start from the edge of the amino acid sequence, and the biologists will have told us what the sequence is. They have sequenced the protein, so this is 1H. We are going to have a signal here, and a signal there, then a signal there, and a signal here, and a signal here, a signal there, and there, and here, and here, and there. So it's not immediately obvious from the picture, but of course there are matching pairs of signals in here. This is because we correlate this proton to that carbon and the preceding one in its own turn with this proton will be correlated to itself, and the next one, and the next one, and the next one. And so you can actually trace the protein connectivity through this picture. And that should be a little bit further up, like that. See? And so that establishes not necessarily which amino acid this is, not necessarily which type or which sequence, but it tells us which peaks in the NMR spectrum correspond to sequentially connected amino acids. So we can say, well, okay, we know which pairs of peaks form a sequence, and then hopefully from extramagnetic resonance data we can later tell by exploring these side chains. So there should be a side chain here as well, what actual types there are, and because we know the sequence we can match those sequences of amino acids that we know to the sequences of these peaks, and that's called sequential assignment. Now notice here as well that we cannot immediately tell which direction is forward down the amino acid chain or backward in this spectrum. And to make sure of that, there's an extra experiment. So that's our item B in part A, in part 2, and that's HNCO, C alpha. And that sort of abbreviation says, okay, we start on the protons, go to the nitrogens, go through the CO carbon into the C alpha. And we detect HN and C alpha, but we make sure that we do pass through the CO on the way. Let us think what that is going to accomplish. So we start like we did on the protons here. So the blue is this HNCO C alpha. We move on to the nitrogen, so like that. But then we are required to pass through the CO. So the sequence takes us to the carboxylic carbon. But then, of course, this CO is only coupled to one C alpha. There is no strong J coupling to this C alpha, and so we will move here. And then eventually, right, use this -8 Hertz to move back to the nitrogen and then back to the proton for detection. And so what is going to happen is this. Only looks at which signal corresponds to the own amino acid rather than on and back. And so what is going to happen here is one of these is going to disappear and be replaced by just one peak per strip. And then we can tell which of these two signals belong to the own amino acid and which belongs to the next, because one of these experiments goes both ways, but the other only goes one way. Okay, making progress. Exactly the same game may be played on the CO instead of C alpha. So this we have discussed. So experiment C is HNCA. HNCO, sorry. Exactly the same principle, but we are going here, 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 and then back. Now that is a local experiment, so there will be one peak per strip, but we can make it non-local by having HNCA, CO. And let's think what that is going to do. So we have H to N. We are required to pass through the C alpha, so it will populate that C alpha and that C alpha. And then we want to end up on the CO. Since there are two C alphas, they will go to their corresponding COs. So we will end up here and here. So once again, this pair of experiments is going to have exactly the same relationship between them as HNCO C alpha had with HNC alpha. One of them will sequentially connect amino acids, and the other will indicate which is the current amino acid and which one is up or down the amino acid chain. So sequential assignment process. Finally, and we will not discuss so E side chain experiments. Those are designed to explore these side chains and to also engage the H alphas. And this is where the specificity kicks in, because different amino acids have different R. H alphas tend to have very distinctive chemical shifts. Likewise for C alphas and particularly C betas, because certain amino acids don't even have a C beta or C alpha. So for example, glycine doesn't have a C beta, proline is cyclic, so doesn't have a C alpha, and so on. So chemical types will then influence the chemical shifts of those H alphas and further protons down the chain. And so it is then possible to tell which amino acid type we are looking at. And then, okay, we now have information. Firstly, about the amino acid sequence that chemists told us about. Secondly, about the peak sequence that we have figured out from the NMR spectrum. And finally, for at least some of those amino acids, we would be able to reliably tell which type they are, because tryptophan is certainly going to look different from valine, right? One being aliphatic, the other being aromatic. And so we know the amino acid sequence, we know the peak sequence, and we know occasionally which peaks are which amino acids, so we can now match the whole thing. And that assigns the entire protein. So this is exceedingly laborious. donkeys years ago in the 90s, assigning a series of protein NMR spectra was a PhD project, and then determining the structure from that. These days it is done in days and occasionally hours, if you are lucky, because instruments got so good and particularly software got so good. So there was a concerted effort by the community called CCP and Collaborative Network for NMR Processing, who have produced brilliant software that does all of these things semi-automatically. Okay, and now to the structure. So the next stage, of course, so item 3, nosy and structure. We have discussed nosy already in the previous lecture. Every cross peak corresponds to a distance. You can be quantitative or semi-quantitative about it. We can of course say that okay, it's 1 over r to the 6, but typically there are various secondary processes like self-relaxation, like cross-relaxation across multiple jumps and so on, that make the nosy volumes only semi-quantitative. So we have peak volume data. So we take a few hundred, or perhaps, you know, over 1,000 occasionally nosy peaks. We can say that, well, okay, approximately they are 1 over r to the 6. So we can classify distances into, for example, long, medium, and short. So we get the distance table. Restraints, they're called. At the same time, we can record things like HSQCs there, and by playing with decouplings, we can measure J-couplings over dihedral angles. So for example, this J-coupling here, if we look at H alpha and its coupling to Hn, so that J-coupling here is going to be a function of the dihedral angle, right, between that bond and that bond. Remember Karplus curves, right? So it had a harmonic dependence on the dihedral angle. If the angle is 0, J-coupling's high. If it's 180 degrees, it is high. If they're at 90 degrees, the orbitals are perpendicular. At least the Fermi contact part of the coupling vanishes and it is reduced. So from J-couplings, by using Karplus curves, we arrive at dihedral angle restraints. Which is useful because that is a constraint on molecular geometry. And finally, what is done is we assemble the amino acid chain inside a computer in a molecular dynamics simulation. So we design our own MD energy, and we have some molecular dynamics specialists in the audience here that know more about the subject than I do. So remember the molecular dynamics energy. We have all of the bonds, and this is not something we need to worry about because we already know which amino acid sequence we have. Then we have the angles. Likewise, those are usually at the sort of energies that protein folding is involved in. That probably doesn't matter either, but dihedrals do. So energy of the dihedrals. This is where our restraints come in, but we look at that. Then we've got charges, right, coulombs. Then all sorts of other things, you know, polarizabilities and so on. You know these things better than I do, but we put extra energy terms in, and those are lambda, so let's make a sum, over all of the distance restraints that we have, some amplitude coefficients, and probably it's just one coefficient in here, so let's put it in front for simplicity, and then we know roughly which distances we have. So we have our R, K, and that is the distance that we have in the system, minus the target distance that we have obtained from the Overhauser effect experiment, and we want the norm squared of that vector with some coefficient that regulates the importance of this term relative to the energies. Likewise, if we have dihedral restraints, then we will have some other coefficient, let's say delta, sum over those dihedral angle restraints, then the amplitude phi k minus phi k 0, suitably turned around to make it periodic, of course, squared, plus any other restraints that we may have. Notice what this does. This updates our molecular dynamics simulation to have the proper molecular energy that will make sure that the molecule stays chemically sensible. However, it introduces artificial energies, which are not strictly speaking energies. They are simply deviations from the distances and the dihedral angles which we expect to find in the molecule based on our NMR data. And so if we minimize this energy, subject to some sensible choice of lambda and delta, we don't want to overdo it because, of course, then the chemical structure will no longer be sensible, but we don't want to underdo it because then the chemical structure would not follow our restraints, right? So this is a penalized optimization with additional terms that enforce the structure that we expect to find based on the NMR data. So we minimize this. And what is the current best way to find the minimum energy structure of a conformationally floppy molecule? Simulated annealing. All right, you heat up the box repeatedly. You slowly cool it down to allow the molecule to settle into the conformation that we would like it to have. And at that point, these restraints serve to guide the molecule to the right place. So we do so repeatedly, sometimes hundreds of times, and that produces not necessarily identically the same structure, because multiple structures may be consistent with the set of the energies and restraints that we have, but we will get a structural ensemble, right? That gets us, well, what our data tells us is the structure. And finally, item 4 is validation. It is known what the distributions of dihedral angles are within the backbones and the side chains. It is known which amino acids are supposed to sit on the surface, typically hydrophilic ones with OH groups and H2 groups and so on, and which inside, so typically aromatic and aliphatic amino acids will be sitting inside. So essentially this is just common sense plus statistics. That tell us that the structure is chemically sensible. Now I'm simplifying massively everywhere here. I've just given you the content of a sizable book in one lecture. A book is cited in your handout, but that's the general idea. So let's just go through the entire thing. Again, we have a class of magnetic resonance experiments. Of course, 4-dimensional and even 5-dimensional ones exist. The trouble with these experiments is they're very long, right? Concentrations are tiny because molecular mass of the protein is huge. So we are talking millimolar concentrations if we are very lucky, and typically, you know, dozens to hundreds of micromolar concentrations, so sensitivity is low. So we need to do multiple repeats on the T3, you know, hundreds of repeats sometimes, to get the decent signal to noise ratio. So an individual readout in T3 is perhaps a couple of minutes if we are lucky, and then that needs to be multiplied by the number of increments in T1 and again by the number of increments in T2. So if we want 256 points there and we want 256 points there, right, 256 times 256 is 16,000. And so if that's a couple of minutes, then welcome to a week-long experiment. And this is why the superconducting magnets need to be so stable. Certain proteins cannot live that long outside the cell, so that makes it difficult. You know, temperature stability, even phase stability of the instrument clock, right? Remember, these are all in specific phases, and the phases are phases of a gigahertz frequency. So if the clock gets it wrong by 10 to the minus 9 in relative terms, then the phase is going to be off. So the stability of the clock of the instrument has to be extraordinary. So all of that only became possible with the latest electronics that in certain cases operates on quantum mechanical limits of what's possible at room temperature. And occasionally people go to cryogenic circuits, cryoprobes, cryogenic detectors, cryogenic amplifiers, and so on. This is extremely sophisticated. Anyway, there's a class of 3D experiments where the abbreviations simply encode the path that the magnetization takes. The letters themselves are the 3 frequency coordinates or chemical shift coordinates eventually that we detect, and whatever occurs in brackets says the magnetization must pass through that atom on its way to some destination. So that's an example of HNCA, literally 3 inept sequences with delays. From protons to nitrogen, evolve on the nitrogen, move to C alpha, evolve on C alpha, move back to the protons through the nitrogen, evolve on the proton. That's our 3 coordinates and therefore 3 chemical shifts. It makes use of this fairly complicated mesh of J couplings, and there are 2 types of experiments. So HNC alpha and HNCO C alpha, one of them goes to the own C alpha and to the previous C alpha. So we have 2 peaks, and that allows us to connect the amino acids, and the other goes just to the own C alpha, and that allows us to tell here which of the 2 peaks is which. A similar experiment is HNCO, which only goes to own CO through this -14 Hz coupling, but HNC alpha CO goes from H to N to the 2 C alphas and then to the 2 COs. So again, sequential connectivity is established. And finally, and I haven't mentioned any of them, there's a large group of side chain experiments that allow us firstly to further assign all of the carbons, protons and nitrogens in the side chain, and secondly to determine the type of the amino acid. So we know the amino acid sequence from the chemists, we know the peak sequence and a few amino acid types from NMR. That's a complete match, at least if you are lucky, and that tells us which signal is which, and there are sometimes thousands of those signals to look at. Once we know which signal is which atom, we take a nosy or nosy HSQC that is selective with respect to each nitrogen and slightly less crowded as a result. We get peak volumes, we convert them into distance restraints. We use extra types of experiments here to precisely measure the J couplings. We know the Karplus curves from other experiments. We get the dihedral restraints. We concoct ourselves a molecular dynamics force field with all the usual terms in it, plus additional energy terms, which aren't really physical energies, but just mathematical deviations from the distances and dihedrals that we expect. We minimize that pseudo energy repeatedly with simulated annealing. We get the protein structure or an ensemble of structures. We send it through validation. Hopefully it passes, and we have our shiny protein structure rotating on the screen. So in the 90s, that's 3 years of work per protein. These days, a couple of months, perhaps, but some proteins don't crystallize, and so this is how it is done. Yeah, any questions?